so vision. Vision is obviously the sense of seeing things. So you have light, and light is going to go into your eyes, and it will strike your photoreceptors, which have visual pigments in them. And those visual pigments are called rhodopsin. Just write that here. And rhodopsin consists of retinol, which comes from vitamin A, and opsin, which is a protein. Now, retinol is going to change shape when photons of light hit it. It's going to go from what we call cis conformation, or you might hear it referred to in the chemistry class as boat conformation. It's just the shape of the molecule itself, so the constituent parts are sticking off on the same side as each other in this particular conformation. So that would be cis or boat. When the photon of light strikes the retinol, one of those constituent parts is going to change and flip over to the other side. So instead of having the constituent parts sitting on the same side, they're going to be on opposite sides. And this would be called trans conformation or chair conformation, because it looks kind of like a little lawn chair or something. So you'll see that sometimes in chemistry with those two different names. We're just going to call it cis and trans. Just cis means same side, trans means opposite side. So in order to understand vision, let's first look at a photoreceptor at rest. So when we're sitting there in the dark, you're not seeing anything, your eyes are closed, you're not seeing anything at all. This is what is going on. And this is going to be kind of interesting because it's a little bit backwards from everything we've ever learned about vision in the past. So no light coming in, we're completely in the dark. Our retinol at rest is in cis conformation. The opsin part of the rhodopsin is inactive because the retinol is in cis conformation. So the opsin's just sitting there. Now, what's going on within the photoreceptor itself, so whether this is a rod or a cone, doesn't matter, it would be the same effect. Rods, by the way, are for black and white vision, and cones are for color, which is easy to remember because there's a C in cones, and that stands for color. But either way, whether this is a rod or a cone, our retinol's in cis conformation, our opsin is inactive, and that allows cGMP to be present in the cell. And we've seen cGMP as a second messenger before, and we know that it stimulates a cell. So the way cGMP stimulates a cell is it holds sodium channels open. When we have sodium channels open, sodium goes into the cell, diffuses along the cell membrane, causing a depolarizing graded potential, or an EPSP. And it depolarizes the membrane all the way to negative 40 millivolts. So more about that in a second. We also know about the sodium-potassium pump that pushes sodium out of the cell while pulling potassium back in. So sodium is getting kicked out of the cell. It's coming into the cell because the sodium channels are held open by cGMP, but then it's getting kicked out of the cell as well by the sodium-potassium pump. So we have sodium going in and we have sodium going out, and it's just sort of cycling. And that's an electrical current that's happening. And it's happening in the dark. You're not seeing anything, so it's called the dark current. Now, this dark current is enough to trigger an action potential in the photoreceptor, so because it's negative 40 millivolts. That's above threshold for this photoreceptor, so it's having an action potential, and because it's having an action potential, it releases the neurotransmitter, which is called glutamate. Okay, so let's follow the rest of the pathway in our minds here. The next cell is a bipolar cell. The next cell after that is a ganglion cell. The axons for that form the optic nerve. And from the optic nerve, we have optic nerves coming out of one eye, optic nerves coming out of the other eye. They cross and make something called the optic chiasm, which means literally cross. Then it goes back to a structure in the brain close to the brain stem called the lateral geniculate, nuclei. And then it radiates back to your visual cortex, which is what receives the sense of vision. So in order to have the sense of vision, you need to have an action potential in the bipolar cell and action potential in the ganglion cell, which will then be carried down through the optic nerves, through into the optic chiasm, to the lateral geniculate nuclei, and then through optic radiation out to the visual cortex, and you saw something. Now, you are not seeing anything here. This is at rest. So think about this. We're at rest. That means we're not having an action potential in the ganglion cell. It means we're not having an action potential in a bipolar cell, but we are having an action potential in the photoreceptor. So what does glutamate do? Glutamate is being released, making us not have an action potential in the bipolar cell so that we don't have an action potential in the ganglion cell. So glutamate then must be inhibiting the bipolar cell, keeping the bipolar cell from having an action potential. This is not 100% understood, so this is a little bit still an area where people are looking into it to try to figure out exactly what's going on, what does cause the action potential in a bipolar cell. We know that we need to stop glutamate release in order to allow the bipolar cell to have an action potential, because the glutamate is inhibiting it. Think IPSP, stopping it from having an action potential. So there are some other cells that may come into play in here, which we'll talk about in a minute, that may assist the bipolar cell in having an action potential. Not 100% sure yet what those are actually doing, but we do know that glutamate is an inhibitory neurotransmitter and that it inhibits the bipolar cell. So that's what's happening when you're in the dark. When you open your eyes to see something, the photon of light comes into your eye. It changes the shape of the retinol to transconformation. Once that retinol changes to transconformation, it activates the opsin. Opsin activates this enzyme here, transducin, and transducin activates phosphodiesterase. And we've seen phosphodiesterase before. We know that it breaks down cGMP. So what cGMP was doing back here, it was holding open sodium channels. So when we break down cGMP, we close our sodium channels, no more sodium coming in, no more dark current. So we can repolarize the photoreceptor, and we'll repolarize to about negative 70 millivolts. What that means is no more glutamate being released, no more inhibition of the bipolar cells, which means that the bipolar cell can go ahead and have an action potential, which will lead to an action potential in the ganglion cell, and that axon will go out through the optic nerve and back through the pathway that we talked about before, where it will be received by your visual cortex, allowing you to see something. So it's a little bit backwards from the way we've talked about neuron function before, in that the neurotransmitter is being released when the cell is not stimulated, and we stop releasing neurotransmitter when the cell is stimulated. Okay, so let's just take a look at these bipolar cells and these ganglion cells, and this cells that might be between the bipolar cells and the ganglion cells. So these cells that we were talking about right here. So one thing we need to think about first are the rods and the cones. We already mentioned that rods are for black and white vision. We know that cones are for color vision. But there are very specific pathways for either one, whether it's a rod or a cone. So here we have a bunch of rods. They have a bipolar cell that they're sharing, and they have a ganglion cell that is in a direct path with these group of rods here. Here we have a cone, just one, maybe a couple, with a bipolar cell and a ganglion cell. Not as much sharing. So not as much convergence on the bipolar cells and the ganglion cells that comes from the cone as we have with the rods. With the rods, we have a lot of convergence on a bipolar cell and a ganglion cell. What this means is that if you see something in black and white vision over here, this ganglion cell can't tell if it came from this one, this one, this one, this one. So it's not very specific. If you see something here, this ganglion cell, it's very easy to pinpoint where that exact stimulus came from. It came from this cone right there. What this does in essence, with black and white vision in your rods, they tend to be more easily stimulated, so you can have lower light levels when you stimulate your rods where a cone requires brighter light. And think about what things look like when you're in bright light and you're making full use of your cones. Colors are very clear, images are very sharp, because you can very specifically pinpoint where a stimulus is coming from. If you think about when you're in a low light situation, like twilight, or you've got the lights dimmed in the house, or whatever, it's just kind of dark, things are sort of fuzzy looking, color's not very bright, everything looks kind of grainy, and that's because you can't tell, like, did it come from here, or here, or here, or here? You've got a little bit more fuzziness going on. Now, the ganglion cells that come from black and white vision are, so coming from the rods, they're called M cells, and the ganglion cells that come from your cone, so the color vision, are called P cells. Now, let's get to the cells that might be interspersed between the photoreceptors, between the bipolar cells, and the ganglion cells. So here's our photoreceptors, a bunch of rods. There's one cone. There's our bipolar cells. This would be the color pathway. This is the black and white pathway. Here's the ganglion cell, so an M cell and a P cell. Now, between the photoreceptors and the bipolar cells, you'll find things called amacrine cells. Amacrine cells may possibly help to stimulate the bipolar cells. This is that part where people are not 100% certain of this yet, but that seems to be the most likely situation, that the amacrine cells are stimulating your bipolar cells. Also, because it's linking a group of photoreceptors together, it's increasing the receptive field for each photoreceptor. So we're kind of making, like, this one is able to be stimulated by a larger area than it would if it didn't have that amacrine cell. Now, between the bipolar cells and the ganglion cells, we have horizontal cells. What these do are they link your color vision pathway to your black and white vision pathway. So they help you if you're in a low light situation. They kind of help to facilitate vision a little bit, especially in terms of color. So think of, when you're in the dark or darkish, a low light situation, and somebody's wearing a bright red sweater, but you can't really see the red that well, but you can still kind of tell that it's red. That's because of the horizontal cell is linking these two pathways. Or in a black and white movie, you can kind of tell when something's like a very bright color, just a little bit, because of that sort of thing. So that's what those horizontal cells are doing. So that is a sense of vision in a nutshell. And our next videos will be about lost my train of thought, about the autonomic nervous system.